Okie dokie, today I want to talk about gene regulation, and so this is basically we have these genes that we've inherited from our parents, but just because we have a gene doesn't mean it's expressed. This is kind of the idea of epistasis. So just because you have a gene, different genes are expressed in different cells, and that's how we get cell differentiation. And that's basically different cells do different jobs. Differentiation. And then epistasis is just genes that you have but aren't expressed. So if you have a gene for something but you don't have that gene turned on and it's not being transcribed and translated to produce that protein, then it won't show up in the phenotype even though you have the gene for it. That would be epistasis. And kind of this happens in like albino people. It's not that you don't have the gene for the pigment, it's just that you don't have the gene that tells the cell to run or transcribe that piece of DNA to produce the protein which makes the pigment. And so the gene's turned off there, and that's kind of one form of gene regulation. In prokaryotes, you have operon systems, and you have inducible and repressible. And then in eukaryotes, it happens a little bit different. And you have enhancers and transcription factors in eukaryotes. So those are kind of different ways in which we control what genes are coded for in a cell, because an eye cell isn't going to need to make as much protein of actin filaments or anything like that as a muscle cell will. And so to get cells to differentiate and do different things, different parts of that DNA code will be read more often and made into protein more often. And that's kind of an intro into gene regulation. I hope that was helpful.